Okay, so let me introduce the next speaker. Cliff Berry is coming to us from the NIH, where he's the chief of the TB research section. Cliff is an example of an investigator who started in very, very basic reductionist science, where he was trained as an organic chemist. And then, for reasons I still try and understand, he went to the Rocky Mountain Labs, which is a section of NIH, of NIAID, where they study all kinds of very interesting pathogens. It's probably, at least up to that point, and even today, is probably this place at NIH where the majority of infectious disease research is conducted. And Cliff went there and worked on chlamydia trachomatis for a few years, and then moved into Mycobacterium tuberculosis at the Rocky Mountain Labs. And then he decided, or it was decided, that he would move to Bethesda and head up the TB research group at Bethesda. And what's been really quite impressive, if you follow Cliff's career, is that he did move from chemistry, and he moved in from chemistry into natural products, and then he started examining how certain antibiotics that already were being used for TB and found that they were acting in a very different fashion than was the dogma in the field. And then he was part of a group that characterized a new antibiotic, a new family of antibiotics, and they've gone on to continue characterizing this group and have started many clinical trials, many of which are in Korea. And as I understand it, Cliff is managing some of these trials, so he's gone kind of the other route from very basic chemistry all the way to clinical medicine and infectious disease. So with that, I'd like to thank Cliff for coming.
Okay, so thanks for that introduction, Dan. It's nice to be back at Berkeley. The first thing you'll notice is that the title that you have in your program is not at all related to the title that you have in front of you today, and that's due to sort of last minute retrospective look at what I was going to talk about today compared to the program and who I thought the audience would be, and deciding that even though Tom Alber had graciously invited me to come, and I love Tom and know his work well, that probably a crystallography and chemistry talk wasn't really the right approach for this audience. So I sort of gave up on what I was initially going to talk about, and instead I'm going to sort of show you some of the translational work we've been doing in imaging and clinical trials in TB drug discovery. And when I think about California, particularly San Francisco, and I think about tuberculosis, of course, California, San Francisco is one of the highest rates of tuberculosis and always in the news for having actual TB cases, which in the US is a relatively rare thing, with only about 20,000 cases or so domestically annually. But I also think about the rich sort of academic history and the leadership that the San Francisco community has always shown in the TB field, and that's Phil Hopewell, for those of you who don't know him, who's one of the sort of leading figures, I think, in TB epidemiology, who's worked in San Francisco and with the cohort of patients who come through San Francisco every year for a long time. But I also think about this man, and this is Robert Louis Stevenson, who's one of my favorites authors, who also lived in San Francisco and in fact sailed from San Francisco to the tropical South Pacific. He looked pretty much like this sort of cachexic, very thin, typical TB patient. TB ultimately claimed his life in the South Pacific where he was buried. But Robert Louis Stevenson has many fantastic quotes, and if you've never read a lot of them, I would strongly encourage you to. And one of my favorite quotes of his is, "Our business in this world is not to succeed, but to continue to fail in good spirits." Now, I don't think he knew he was talking about TB drug discovery at that point in his life, but in reality he was, because really it's a daunting task, and it's a really tough organism that combines all the challenges of infectious disease with all the challenges of oncology in designing therapeutic strategies for it. So you know the background. I'm not going to take you through a lot of this. TB is a devastating disease. It remains a devastating disease, and drug resistance is increasing. It's a simple message, but as I have personally experienced and have personally dealt with many TB patients in different parts of the world, I just want to put some faces on that instead of just giving you the statistics. So this is a young girl who was a computer science major at Seoul National University, was a patient of ours who had XDR TB as a primary contact case. So probably from the subway system in Seoul, acquired XDR TB and died from it at a very young age. This is another patient of ours in Kabul, Afghanistan. She's jaundiced. She's being treated with isoniazid. She also had multi-drug resistant disease. And these are two more patients of ours who were HIV-TB co-infected from Nigeria, all with actual disease. We have made a practice over the last 15 years of trying to engage directly with the TB control infrastructure and with TB patients and clinical management of TB patients, and use that to try to inform our basic research agenda. So one of the things I tell every postdoc who comes into the group when they start with was, I don't really care what your project is. It's got to be, it's got to be smart. It's got to have a good hypothesis, et cetera. But you have to be able to explain to a patient why this is important to them and what you're doing is actually going to affect what that patient experiences. And so why is TB still such a problem? We have highly effective chemotherapy. In fact, in controlled clinical trials, we can cure 95% of TB patients in their first episode with tuberculosis. And the reason it's such a problem globally is because it takes 6 to 8 months of chemotherapy with 4 drugs in order to cure somebody of tuberculosis. And in the real world, and this again shows some settings where we've worked, a rural community in Cambodia and the Pakistan-Afghanistan border and refugee camps, it's impossible to keep people in treatment for 6 to 8 months and convince them that they actually need to finish 6 to 8 months of treatment when they feel better in only a couple of months. And so a lot of our research has been focused on understanding why TB chemotherapy takes that long. And I think from a sort of conceptual, simple level, it's helpful to sort of see what TB really looks like. This is an upper lobectomy from a 19-year-old kid in South Korea. He's got multi-drug resistant disease, but that doesn't really matter because the severity of disease is pretty similar between drug susceptible and drug resistant patients. And what you see is it's a mess. This is a cavity. Here's a caseous necrotic lesion. This is lots of fibrosis. This is the other half of the cavity. It's just been sliced down the middle like this by my pathologist. And you see a whole constellation of different kinds of lesions. Now, within those lesions, if you're thinking like with your microbiology hat on, the bacteria within those lesions experience very different environments, and they're under very different stresses and very different growth rates and conditions, perhaps nutrients available to them, lots of things different between an aerobic inside of a cavity and the inside of a caseous necrotic lesion that's basically completely hypoxic. So over the last 10 years, I like to sort of, and again, this is kind of artifice, but it's useful artifice for this talk. We've been trying to sort of build a pipeline from the hood to the bedside. Now, you'll notice I said hood, right? That's because at the core of it all, you have to remember I'm just a chemist. I pretend to be a lot of other things, but I'm really just a chemist. And so we'll look at developing ways, and about a third of my group still works actively in medicinal chemistry, not necessarily on this scaffold of compounds. This is PA824, which is currently in phase 2 trials we contributed to. But we also look at a lot of more predictive animal models, and we do a lot of phase 2 studies because I'm quite firmly, and I think in concert with the message that Julie just showed you, I'm firmly of the opinion that we don't learn a thing until we actually get into humans, particularly with regards to chemotherapy and TB. In fact, every one of the clinical trials that we've participated in so far has had the distinction of, I think, being completely wrong from the hypothesis we went into it, but learning something really important by the end about the biochemistry or the chemotherapy of TB at the end. So what I was going to talk to you about today, because it was Tom who invited me, and we had good crystallography data, was this part of the pathway. After last night's plane ride and rewriting the trip, what I'm actually going to talk to you about is the final part of the pathway with just a little bit of a nod towards the predictive animal models. So what are we trying to accomplish in our phase 2 program? Well, typical phase 2 trials at least in TB, are based on microbiology. You'll see people present sputum, culture, and smear conversion rates. And that's pretty much all you learn from TB clinical trials. The clinical symptom scores are not very good. They're subjective, not very quantitative. And the result of this is that you reduce patients to a sort of single outcome measure. They either improve, like this guy, or they don't, like this guy. And so we've been trying to understand and really develop methodology for getting richer and more useful data out of phase 2 that gives us more endpoints than just somebody got better or they didn't microbiologically. So phase 2 ultimately exists to sort of learn how to do your phase 3. So you've always got to keep in the back of your mind, what's the phase 3 program going to look like? And at the moment, you don't need to read the details of the slide. At the moment, the only sort of accepted standard for TB chemotherapy in phase 3 is relapse after discontinuation of chemotherapy, 2 years after discontinuation of chemotherapy. So that's the only thing that the FDA accepts as an endpoint for a phase 3 clinical trial. Now, this is one of the TBTC studies. This is one of the largest domestic groups that does clinical trials in tuberculosis control based at the CDC. And this is one of their studies in which 1,004 patients were enrolled, followed with a particular combination. The details don't matter here so much. What matters is that the only things in the end that they come up that are correlated with relapse are cavities and a positive sputum culture at 2 months. Now, both those look impressive. Cavities has a p-value of 0.001 in multivariate analysis, and the sputum culture is even better than that in this particular cohort of patients. The problem is that's very cohort specific, and in a sort of meta-analysis of 15 big studies involving about 7,000 participants. If you plot the log's odd ratio of positive culture results against the odds ratio of a poor outcome, what you see is at no time, 1 month, 2 months, 3 months, or 4 months, are they really well correlated. So what's happening in the sputum microbiology doesn't necessarily tell you what's happening in terms of clinical outcome for patients on clinical studies. Even though in individual studies between individual groups of people, that correlation can appear quite significant. So what I'm going to tell you about today is several studies, but none of them really in painful detail, so don't worry. But that involve a lot of use of non-destructive in vivo whole body imaging technologies. And really, over the last, again, 5 years, we've tried to understand the dynamics of the response of TB to chemotherapy and how we sort of can underpin that with the knowledge of the pathology and the animal models we have to translate that into better and better phase 2 methodology. And so the two techniques, computed tomography is easy. This is just fancy X-ray images, and I'll show you a lot of them. And this is really structural imaging. It asks what's happening with the pathology of the disease in the lungs. PET, which is positron emission tomography, is radioactive imaging of a probe. And now typically this is widely used in cancer, and all they use is 2-deoxyfluoroglucose. And so it's measuring metabolic activity in a way. People use it as a marker of inflammation very often. And so FDG is trapped, it's phosphorylated and trapped inside cells that are burning a lot of glucose. And so you see an accumulation of signal in cells that are metabolically active or areas of active inflammation. Again, it's widely used in cancer chemotherapy. The uptake is measured in this unit called standardized uptake value, which is just how much radioactivity, how many radioactive emissions are happening compared to the weight of the patient. And this is functional imaging. This is telling you something about how the immune system is actually seeing the disease. So I'm going to break the bulk of the talk now into 5 different sort of vignettes. The first is going to be to walk you through some of the structural features of adult pulmonary tuberculosis, then to show you some of the functional features, and then to talk about the dynamics of the response to chemotherapy and how you can actually look at that, modeling that response a bit in animals, then ultimately looking at what's our endpoint here, the anatomy of relapse. So to start, structural features of TB. And the easiest way to show you this is just to walk you through somebody's lungs who's got tuberculosis. And so this is a series of images of high-resolution CT from a patient with tuberculosis. And you can start to see, I'll stop it at several points. Here's a large nodule that's due to his disease. This is this is not a far advanced case, even though you'll see a very significant amount of disease. Lots of big nodules, these loose areas of infiltrate, this big area of consolidation. And then down even farther, you can see extensive disease in the right lung. So I'll just stop it there and point out a couple of features. It's not atypical at all to see these heavy areas of consolidation. They're not projecting as at the highest resolution, but you can see this is all just small areas of infiltration of nodules, of collapsed lobes. This is actually the collapse of the middle lobe of the lung. So the lung is 3 bags of air. The middle one has collapsed because there's so much infiltration in it, and the other 2 have expanded. And this is, again, this is a pretty modest case of TB. This is not a severe case at all. And as you walk down through that patient lungs, again, you get the sense that there's a spectrum of lesions. There's all kinds of things that are happening in there, and the TB within those lesions does not respond with equal rates in any particular sort of these lesions with respect to different kinds of chemotherapy. Now, that's not a new idea. In fact, that idea that TB is in different microenvironments, that we have to attack those different microenvironments, was first sort of laid out by Denny Mitchison of the British Medical Research Council in the pivotal trials that defined what we use now as first-line chemotherapy for tuberculosis. Now, and Denny's a good friend of mine, and in conversation since then, he said, well, we had the idea right, but we had all the details wrong at the time. And so we've spent a lot of time as a field grappling with what are the details, what do those different microenvironments look like, and what do the individual regimen components actually contribute to that kind of cure rate. And so, again, I'm not going to belabor the point. I'm not a radiologist, although I've learned enough that I think I could play one on TV convincingly. But the radiologists have a vocabulary to describe tuberculosis lesions. And TB lesions come in lots of different flavors. And so these are all the radiologic findings that would be associated with TB. They're things like centrilobular nodules or branching linear structures, tree and bud appearance, cavities, consolidations, et cetera, et cetera. And if you look across TB patients, you can start to get a sense of which of these lesions are present in which patients and which ones ultimately fail from chemotherapy and which ones succeed. And in fact, in this particular study from the Japanese group, from one of the Japanese radiology groups, they defined a system for characterizing active versus inactive disease, which often is a problem when somebody shows radiographic abnormalities but doesn't have sputum culturable bacteria presence. So, it's important to get just a little bit of perspective, and I'm not going to go through all those radiologic features, I promise, but just a couple of them to highlight a couple of points. There are 10 different flavors of active TB in lesions. And you have to sort of imagine, so I showed you a 3-dimensional representation, but it's really hard from a 2-dimensional slice to think about these things 3-dimensionally. But remember, the lung terminates in lobules. The end unit of a lung is a volume of open air that's a lobule of the lung, where the air oxygen is exchanged into the blood. When those lobules fill up, they give a very characteristic appearance like this. And when you've got pus down at the bottom of your airways, which is often how TB appears, then you get these sort of centrilobular nodules, these nodules in the middle of a lobule. In contrast to that, you can have miliary nodules that are scattered across the lung that aren't particularly centered in the distal parts of the lung. This tends to be a feature of early disease, and it tends to resolve pretty quickly, whereas areas of consolidation like this tend to resolve very slowly. There's also big nodules, and these are a bugaboo because you can never be certain they're due to TB, but here's macro or acinar nodules that are present in the parenchyma of the lung. Again, these are mostly solitary things, and it's difficult to sort of, again, think about this in 3 dimensions because these are not isolated structures. They exist in a bag of air in the lung with little bits of tissue connecting them. There's also things called tree and bud opacities, which are these, which have the appearance of a tree with a bud on the end of that lobule. And so again, just not for the details of this, but to understand how different the bacteria within those lesions you might expect the metabolism to be. Cavities are a prominent diagnostic feature of tuberculosis. This is a cavity right here. It's a hole. It can be thick-walled, not like this one. This is pretty thin-walled. It can be spiculated, showing signs of activity as the bacteria are drawing in more and more inflammatory cells. And that's thought to correlate with high amounts of bacterial activity. In fact, the sort of dogma in the field is that transmissible bacteria are generated at the surface of the cavity by active replication aerobically, and then they're coughed up into the airway in patients to infect the next group of patients. Well, across the studies that we've done anyways, we've adopted two different ways of kind of trying to understand what's happening with the CT patterns. The first is a sort of quantitative methods where we do readers studies, and this is borrowing from oncology. And so we'll take a group of patients, I'll show you data from this group of 30 patients, and we'll send them out to 3 blinded radiologists, and we'll ask them to sort of use a semi-quantitative scoring system and score them in all those 10 different categories. Well, the first thing you learn when you do studies like that is that the radiologists don't really, they have all these nice categories, but they don't really agree on what those categories mean. And in some of the categories where it sounds really soft, like tree and bud opacities or ground glass opacities, it really is really soft. All 3 radiologists will disagree on when a patient actually has that. And so it's the same, oncology learned this lesson 10, 15, 20 years ago, and I think they've gotten around it by using increasing numbers of readers and narrowing their definition of what features look like, but TB hasn't had that level of experience. But nonetheless, when you do that, you do get a sense of how people respond. And so this is a set of, this is a single patient. The blue bars are his entry scans, the orange bars are his 2-month scans, and the green bars are his 6-month scans. It's read across 10 different features. These are the different kinds of features. These are the average of the 3 radiologist scores with their standard deviations. And you see they do quite good on some things. I mean, anybody can look at a small nodule that's less than 2 millimeters and say that's a small nodule, so I can semi-quantitatively estimate that. On the other hand, things like bronchial thickening is the standard deviation approaches the measurement, and so it's not as useful to compare those. We've also taken a computational approach to this, so I have a small group of programmers who do image analysis software now, believe it or not. And they've written algorithms to take CT scans like these. This is an actual patient CT scan, sequentially strip off different layers, including the pleural surface of the lung, isolate lungs from those, and then you can ask what's not lung in that, right? So it's pretty simple analysis when it comes down to it. But TB patients have a host of unique characteristics, and it's really defining what's left from that becomes the difficult problem. So here's a patient, high-resolution CT scans, extracted and then colored just by Hounsfield intensity. So that's the measurement of how dense tissue is. So bone is very dense, has a very high Hounsfield intensity, Hounsfield absorbance. Soft tissue is very light and doesn't. And so typical lungs would be completely full of just this sort of gray level stuff. This is a patient with rather extensive bilateral disease. He's got disease on both sides of the lung. He's got quite a few features at high Hounsfield intensity, and that's pretty typical for what we see in TB patients. Again, we can do increasingly fancy analysis of this. I'm not going to take you through a lot of it, but this is again illustrating bronchial thickening in a patient with TB, cavities, very, very dense structures in the apex of the lung. This is typically where you see very dense features. It's often where you see big cavities, and it's often where you see relapse in patients. We can also fit this data semi-quantitatively. We can reconstruct the lungs in patients and track lesions over time with that kind of software. Now, the reason I'm telling you that, in part, is because we operate under the assumption that reader studies are pretty expensive, difficult, and outside of a dedicated research facility like NIH, probably not portable for methodology for clinical trials for TB. Open source code for analysis of radiology data like we're doing for this is something that will be instantly available to the community to use field tests and actually try to implement in clinical trials that other people do outside of the NIH research community. So how does the computer do when we do that? So here's one comparison. This is less than 2 millimeter anomalies. This is characterized by the 3 radiologists that I showed you before, about a 50% decrease at 6 months, no change at 2 months. This is the actual extracted volume in cubic millimeters, 8,000 cubic millimeters of volume at entry, about the same at 2 months, and about 50% reduced at 6 months. So we can, at least with some of those features, very accurately capture information that measures quantitatively the degree of change in patients' lungs over time. And if you, this is sort of a truth in advertising slide. If you compare the volume of abnormal lung extracted computationally with the semi-quantitative score from the radiologists, you get, I think, what is an encouraging trend, but not a very convincing trend. And the reason is this is a lot of those soft features. So the things that are off the line here are the things that are not easily quantitated by the radiologists, nor are they easily captured in image analysis software. So now let me show you some of the functional features. This is, again, one of the patients in a clinical study who was getting a baseline PET CT scan. So patients in this study get a bolus of about 10 millicuries of F18 labeled FDG They get an hour to rest. They void the majority of the radioactivity, so he hasn't completely voided. You can see some in his kidneys here. His heart is lit up, which is an indication of how glycolytic that patient is, so it's a very variable characteristic. And then this is his tuberculosis disease. And you can see there's a fairly extensive amount of disease. There's a lot of inflammation happening in that patient, and that inflammation is a consequence of the infection process that's going on. And so that would be nice, that's one nice surrogate. It's easily quantified, and it's the kind of thing that you can reduce to numbers pretty easily. So what happens over time? And I have to show you one more detailed study to actually tell you that. So here's a patient in another study of ours. This is a study in chronic XDR patients. He has a 10-year history of failing TB treatment. We pick him up in late 2009. His drug susceptibility for his organism is basically pan-resistant. Every drug we have available to treat him with, and a marginal susceptibility to pyrazinamide is the only thing he's susceptible with. So this is a chronic failure case, and we have a lot of these in areas where we treat a lot of MDR-TB. We see a lot of highly drug-resistant strains like this. So in this particular study, patients are randomized to either start therapy immediately with a single agent. It doesn't matter, but it's linezolid, but it doesn't matter for the purposes of this discussion, or to have a 2-month delay in start so we get a hospitalization effect. So this T minus 2 months means effectively nothing changed in terms of his chemotherapy or lack of chemotherapy, because he's only receiving sort of prophylactic things we know he's already resistant to. And in that 2-month time period, this is what the repeatability of the SUV measurement looks like. Now, what you notice is that in general, the pattern looks pretty much the same, but in one particular case, pericardially, this is his heart again, there's development of a brand new lesion there. And that's real. I mean, TB does, one of the things I've come to appreciate with these scans is that there's a dynamic nature to this disease. It changes on a month time course. You can see lesions come and go naturally in people, not the result of chemotherapy, come and go in different parts of the lung as just part of the natural variation that happens. This patient went from smear 2+, which is a lot in his smear, to smear 3+ at 0 months. He went from culture 28 days to positive to 15 days to positive, which means he's more bacteremic at 0 months than when he started the study. And 6 months later, on monotherapy with a single agent with XDR disease, he's completely clean. And that correlates with a complete conversion to smear and culture. So first message, it's a great drug. Of course, it has its own problems. Second message, though, you can quantitatively measure the effect of the drug in a relatively short period of time. Now, that's 6 months. That's not relatively short. But in fact, we can look much earlier than that, and I'll show you that in just a minute. What does it look like on the CT? So in the CT, at 0 months, you can see the aggravation that happens in this patient. This area looks denser, it looks more angry, more active, and this pericardial lesion I was showing you on the PET scan actually pops up. It comes from nowhere. It wasn't there at baseline, and it isn't there, and it was there at 0 months. At 6 months, that lesion's resolved, and these lesions have gone a good way towards resolving. This is about as good as you ever do for very thick cavities. They never completely disappear. They collapse into these sort of bronchiectasis-like structures. So what's the lesion, what's the PET really telling us about what's happening in the lesions? And the answer is, I think what it's telling us is reporting on the global severity of the disease. If I track the SUV maximum per zone in patients, and this is across 30 patients, with the severity as read by our computational algorithms, I get a half-decent correlation between the severity of the disease and the absolute maximum intensity of the radioactivity from the fluorinated glucose. In other words, there's the most inflammation with more disease. It's not rocket science, but it's important to establish. So then, how can we use this to look more closely at the dynamics of the response to therapy and what actually happens in response to therapy? So here's one patient, and again, I'm going to fall back to the sort of standard methodology. This is the structural analysis. So these are HRCT scans collected at 0, 2, and 6 months on effective chemotherapy. At 2 months, there's really no change in this patient in terms of the structure. So the amount of destruction that's happened in his lungs is pretty much the same, and you've seen that measured before, and the measurements are plus or minus 10% the same structurally. By 6 months, he's clearly getting a lot better. You can see even some of the airways have started to sort of reopen and regenerate, and there's much less disease in that patient at 6 months. And CT is known to give you a pretty good indication of how people are doing at 6 months. It's one of the fundamental ways we can measure cure in patients. And we can measure all those specific features. This is the radiologist's measurement of those features. So small nodules, 50% reduction. Tree and bud opacities, perhaps 60% reduction. Cavities, they can't really agree on. And large nodules don't change at all, even over 6 months period of time. And now if you sort of take a step back from that data and say, okay, here's a bunch of data sets. Here's 30 different patients. Here's their score. And this is the less than 2 millimeter nodule class. And if you squint at this, it's particularly helpful if you've had a glass of wine or two before you do this exercise. You can start to see this is a combination of perhaps 3 different kinds of patterns of response to therapy. People who respond fast, people who respond slow but still respond, and people who don't respond at all. And if you just take that sort of simplistic approach to that analysis, and you apply it to all those categories in a whole group of patients, and you ask what categories do people get better in faster, what I can say is that this is the change in entry minus 6-month HRCT. These are people who get better than 50% better. These are ones who don't change much, and these are ones who get better than 50% worse. And then I can take my patients and line them up based on their clinical response and their microbiological response. So this is sort of a rank order in this group of patients for who did well to who did very badly. In fact, the bottom 3 patients died during this study, so this is the ultimate bad, and they got worse in every single characteristic. Then you start to get a pattern of response in different features that shows you things like consolidations and tree and bud opacities in general respond pretty quickly and are dealt with pretty effectively by this particular combination of chemotherapy. For this talk, it doesn't matter what that is, but this is a particular set of chemotherapy, a particular type of chemotherapy with sensitive and resistant patients on the top and bottom. Whereas things like bronchial thickening, ground glass opacities, collapse in particular, and even cavities to some extent don't tend to get better as fast. Well, there's a lot more information in that, and I'm not really going to give you a physics lesson, but I want to sort of give you an appreciation of how we're trying to do that analysis. I've introduced you before to the concept of Hounsfield units. So here's some basics. Air is minus 1,000, fat is minus 120, water is 0, muscle plus 40, contrast agent is about 130, and bone is about plus 400. When you're doing these sort of high-res CT scans and you're looking at them these sort of from a computational perspective, you can start to ask about individual lesions. What happens to individual lesions on chemotherapy? And so here's one lesion within a patient, isolated by the computer with a heat map on it that's imposed by the absolute Hounsfield intensity. And so you see this is kind of a uniform picture. It's kind of a soft, gooey ball of fat in the middle with a kind of a muscly surface to it. At 2 months, it looks roughly the same, but it's starting to develop a hole in the middle of it. And at 6 months, as the disease is actually resolving, this hole is becoming bigger, and this is turning into what you might be tempted to call a cavity. But in fact, that's the way the lung reorders itself, and that's the way it reorganizes itself into its normal structure. Lungs air, so when lesions resolve, they go back into basically air with thin interstitial tissue. So here's another cavity in this, or another series like that. In this particular one, I'm going to move back in the Z-plane and show you what happens inside the same lesion over time on treatment. And again, as you go through the 0, 2, and 6 months of chemotherapy, what happens is the lesions tend to dissolve from the inside out as the inflammatory cells move the disease back out of that lesion and move it back into normal lung architecture. And so as you watch through the middle of these, you then get a sense of what response you would see in different patients and what kinds of responses are favorable for healing and what aren't favorable for healing. But what about the PET scans? That's what I really wanted to tell you about. And so here's that same patient you looked at before. This is his baseline scan. This is his heart, which remember, just ignore from this picture. I should just cut it out, but it feels kind of wrong. And then this is his PET scan after 2 months of treatment, a time when there's no change at all in the CT. The structural imaging hasn't changed at all. And if I just take a surface across this lung and show you what the maximum intensity of the radiation is, you can see at baseline it's got this intensity, and at 2 months it's got this intensity. So it has, in general, lower intensity, but there's one particular area where it's higher intensity. If instead I look at just the average SUV, which some radiologists like to do, then what you see is just a general decrease in the uptake at 2 months. Again, an indication that the patient's getting better, but at 2 months in the CT, you couldn't tell that. So it's a surrogate for the 6-month CT response. But what's really happening with this lesion, because that's the interesting thing and the reason I picked this patient out of the lot to show you again. This patient actually has a lesion at the region of collapse of his middle lobe that actually increases in intensity. So there's an increased, heightened inflammatory response in that particular kind of lesion at 2 months of chemotherapy. And it turns out, through imaging hundreds of patients now and looking at the dynamics of that, that's not at all uncommon, particularly in collapses as they start to resolve. And you can imagine what might happen is the bacterial products are released from those masses of bacteria that are trapped by the collapsed lung tissue, and you're getting more inflammatory cells recruited by more and more bacterial products that are pumped out into the local environment of the lung. So can you really use it as an early endpoint? Because what we're ultimately trying to do is to do our phase 2s where we're getting this information from single agents and we're doing it at a very early time point. So here's another patient with a very long history of treatment with disease. This is a 28-year-old male who started in 2001 with drug-susceptible disease, was cured, treated again in 2003 when he relapsed, failed multiple different rounds of second-line chemotherapy. And when we picked him up, his drug susceptibility test was resistant to these 10 agents and supposedly sensitive to kanamycin and capreomycin, although he'd been clinically treated with them for over a year and hadn't responded at all. So again, this is a chronic failure case. You can see he has actually quite localized disease. There's not a huge amount of disease in this patient. His heart is bright, and this is not particularly hot radioactively, but he's got these lesions in the upper lung that are making it into his airways in some numbers, because we can see it on the smear and we can count it in days to positivity in the BACTEC culture. Now, after only 1 month of treatment, what you can see is that he's converted to smear negative and 37 days to positive, which is indicative of a positive effect in this patient. But you can also see that the CT or the PET image is the PET signal is dramatically reduced. Again, you'd have a difficult time telling that from the CT pictures. So here's the CT at 0 and at 1 month, and here's the PET intensity, 5.6 is SUV max here. Here it's 2.5, reduced by about 50%. So again, we're trying to develop this as a indirect, preliminary sort of indication of how a particular chemotherapy would work, so that you could benchmark 6 or 7 things to say which one agent do we want to actually take forward into phase 2 or phase 3. I'll skip that. It's just more showing you even that this quantitative nature of it, a 50% reduction, is true in at least 10 different lesions in that patient. And so it's not just the phenomena of the one that I picked. So that's nice, but ultimately human studies, even though they teach you important things, lack the ability to teach you what that means in terms of the dynamics of what's happening to the bacteria in those lesions. And so in order to do that, you have to be able to either sacrifice animals or figure out a way to get bacteria out of people in a way that lets you understand what's happening to the bacterial populations. And so we've been working to develop better models for modeling the human response to animals and really benchmark those models against the human chemotherapy trials that we've been doing. And one of the challenges in this is that despite the widespread use of mice as a TB chemotherapy model, mice don't develop any of the pathology that I've shown you. And so mice would be sort of a 0 on the radiologist scale, even at baseline. They develop a sort of diffuse pneumonia, but none of the characteristic pathology. And so we really had to go to higher vertebrate animal models to see this kinds of pathology. This is a fused PET CT scanner that we designed and built inside of our BSL-3 suite. I should say we did this in collaboration with Joanne Flynn at the University of Pittsburgh, our engineering group at NIH, the Siemens engineering group, and this group, this human brain scanner, the Seratom group, all of which was a huge feat and partly funded by the Bill and Melinda Gates Foundation. What we managed to do was take a registered bed that slides the animal through the gantry of the PET scanner and the CT scanner and collect registered images from medium-sized animals. Now, this is a cynomolgus macaque. This is a rabbit. And I'm going to show you studies that involve neither of those two particular animals, but we've done lots of PET CT and lots of chemotherapy trials in those. But my favorite animal model by far is the common marmoset. Now, again, to understand why it's my favorite, you're going to have to remember that I'm a chemist at the heart of it, right? So the biggest thing about marmosets is they're small. These are the size of laboratory rats. So they're 250 grams as adults. They're tiny little monkeys that look and act like humans, sometimes scarily much like humans. And this particular animal illustrates, this particular set illustrates another striking thing about marmosets. 80% of live births are twins. And so because of that, every time we infect a set of twins, we've got a matched control and drug treatment animal for every single one that's PK matched. So we don't have to worry about PK. Now, the immunologists get excited about this for different reasons, but we've developed it as a chemotherapy model only. And so this particular set of animals was developed from a clinical isolate from a patient of ours in South Korea. These two animals received at the same time a relatively high-dose aerosol infection, and they developed a very scarily similar picture of disease, 83 and 82 different lesions that was indistinguishable even by my pathologist from human tuberculosis. We had cavities, we had the entire spectrum of different lesion types that I've shown you in the human disease inside of a marmoset model, and we could capture effectively high-resolution PET-CT images of these animals. Patients. I believe they get TV time every day, so sometimes they feel more like patients than animals. I mean, they're beautiful animals that it's hard to do this with. But these animals actually, again, develop very similar disease. This is an animal 40 days post-infection. And then what we wanted to ask was whether they resolved the infection with kinetics that were similar to human disease and with standard frontline chemotherapy. So we didn't want to use our experimental agents to benchmark it. So this is what every TB patient under the sun gets with their first presentation with TB. Isoniazid, rifampicin, pyrazinamide, and ethambutol. 4 drugs in combination immediately and upfront. And with drug-susceptible disease, most patients respond really well to that, right? So this animal was given that at a human PK equivalent dose. The other thing I haven't mentioned about these is that they're accepted by FDA as a surrogate PK marker species because their PK in general, their metabolism of drugs tend to be very similar to humans. So all you have to do is allometrically scale these is weight by weight, and you tend to get very similar PK profiles in these animals. By 14 days of chemotherapy, there's a dramatic reduction in the PET signal. You can sort of see from the diffuse blue pattern on the background that there's really not much change structurally in the CT image. And by 28 days of therapy, it's virtually gone. So that's highly effective chemotherapy, a very good surrogate marker, even at 14 days of chemotherapy for telling that it's highly effective chemotherapy and in a very relevant animal model. So how does that compare to a human? So here's a human scan. This is a patient of ours in Stellenbosch in South Africa who's got a PET-CT. This is a drug-susceptible patient. He, on entry to this, had extensive disease. It wasn't bilateral, thankfully. There's not much destruction going on in the right lung. It's all localized to the left lung. And here, on the same regimen as the animals were getting, HRZE at study entry before he'd received a single dose, and 28 days later, a dramatic reduction in the PET intensity. No changes, by the way, in his CT picture. So again, it's supporting, we're trying to build this case. Ultimately, we need a lot more evidence and a lot more patient scan that this really may provide an indicator of early response to chemotherapy that we can use in phase 2 studies. So the last thing I want to show you is just an example and inferring a lot from an example. But this is a patient, this is one of the things that we're good at, which is following patients for a very long period of time. So this is a patient who came to us in 2006. Actually, I remember the day, this was the day before Christmas that we enrolled him in study 2006, and I was present for that enrollment. We started him and have followed him over a course of several years with sequential imaging. He initially started on experimental chemotherapy. He didn't respond all that well to it. He responded okay, but not that fantastic. And these are what his PET-CT images look like. So in September of 2007, when he was basically sputum culture negative, there was still residual uptake in the PET-CT. And we've seen this in about a dozen patients out of 150 that we've looked at so far. There is still residual impact. There's residual amounts of disease even after 6 months of chemotherapy. Now, I haven't shown you his baseline scans, which were horrible, I'll tell you. I mean, he had very extensive bilateral disease and a lot of bilateral apical disease, which I've come to recognize is a surefire sign that the patients aren't really going to do that well in the end. The way this works is they get second-line chemotherapy until they convert their sputum, and then they get 1.5 years more chemotherapy, and then we follow them up for another 2 years after that and take scans throughout that period of time. And so this patient in November of 2007 was discontinued from his chemotherapy because he met the 18-month threshold, which is the WHO-accepted MDR treatment regimen, 18 months after you've converted your sputum. Despite that, there's a little bit of residual activity. Now, in March 2010, he's coming back for a well visit, a follow-up visit, and what we saw was inflammation back at the initial sites of his disease. This is where it was, this is where it is in March 2010. Now, at that point, his bacteriology was negative. He was not formally a relapse. He had no symptoms, he was well, he was apparently still cured, but the PET scan was telling us that something was going on here. In fact, it was only a few months later, in May of 2010, that he came back smear and culture positive again, so he was a formal relapse case. And went on more recently over the course of this last year to be initiated on a third round of our best guess at what the chemotherapy should be for this kind of patient. So this is what a relapse really looks like. Now, you might be saying in the back of your mind that that's not so surprising. He had extensive disease and it came back at the site of extensive disease. But in fact, that's been highly contentious because people say that the bacteria, the latent form of the bacteria, hides out in somewhere, like in the lymph nodes, or it hides out in the fat or the skin or somewhere else. Well, we've got about a dozen cases that look like this now. It's not hiding out somewhere. It's at the site where it was initially, and that's where the disease is going to relapse. And I think that's an important appreciation of like the physiology of what we can expect those bacteria to be doing. They're not magically turned into spores and living in the lymph nodes. They're actually just bacteria that persist despite a lot of chemotherapy. So in conclusion, there are sort of quantitative measures of lung pathology that you can apply to TB patients and that are superior to sort of conventional quantitative radiology. We think this dynamic of functional imaging really adds a lot to the way we look at TB patients. And in a whole group of studies I haven't told you about at all, we're trying to develop bacteria-specific PET probes. So we're not just able to look at inflammation, but we're able to look at aspects of bacterial metabolism. And I know several other groups are doing similar projects. But these imaging techniques really do add a new sort of quantitative measure to endpoints in clinical trials. And we think these higher vertebrate animals can be used ultimately to really pilot what our phase 2 and phase 3 trial design looks like. And the last message is cavities are responsible for relapse. And by the way, mice don't actually get cavities. Okay, so I've shown you a lot of work, most of which came from my clinical group, but a lot of the basic science happens in my group in Bethesda. There's a bunch of, these are stratified by discipline, so the chemists are in front, the biologists in the back, and the clinical people at the very farthest part of the apex. And yes, I forced them all to wear black for it. And then that, of course, is mirrored by my clinical group, which is primarily in South Korea, although we do have sites in China and South Africa that we work with as well. This is a joint partnership between the Ministry of Health and Welfare of South Korea and my institute, NIAID. And we've built a research lab and a BSL-3 facility, and we have about 30 clinical research staff there who are shown at we have annual group meetings. They tend to be in Korea every year. This is the 8th annual retreat and symposium that we have. We welcome lots of outside participation in this. We invite our biggest critics and our best friends usually and have a round review of every project that's going on with it. And this is, you'll recognize some of the same faces from, well, that's not Rachel, that's Rachel. From site to site, because I bring most of my lab from Bethesda to this, so they meet patients. And again, ask the sort of acid test question of their project. Tell him why your project's important and why what you're doing matters to the patients in South Korea. It's been a great 10-year odyssey of working with the South Koreans in developing this joint center. I have a great number of wonderful friends, dedicated scientists and clinicians. These are two of my lead clinicians, Sunny Lee and So-Young Lee, who take care of all of those highly drug-resistant patients. And then it takes a huge clinical infrastructure staff and a huge amount of patience to deal with the bureaucracy associated with doing things in clinical trials. But I won't say any more about that. I'll just say it's worth every cent and every ounce of effort you put into it, because at the end of the day, you learn something real about the disease and not just something useful in the laboratory. Thank you very much.
How do you know for a fact that one relapse patient, it was the same bug?
So that's easy. I mean, we have isolates from every point in their treatment. And so it types, in fact, I think we've actually whole genome sequenced that particular isolate from his initial presentation and from his relapse case.
Do you get people who get reinfected with different isolates after treatment?
Sure, yeah, yeah. And that's easy to recognize, though, by either molecular typing methods or more and more genome sequencing's got cheap enough that we tend to just sequence the whole genome of any isolate we get from a patient.
Hey, Mark?
You suggested that one of the reasons why treatment was so difficult was the many different environments that get established in the example you showed of lung. Is that because it's difficult for drugs to penetrate these environments, or is that because the physiology of the bugs is different depending upon the environment?
Yeah, it's a great question. I wish I knew the answer to it. So we actually have two studies going on that address that. One is a quantitative high-throughput screening set against whole organisms, where we've changed the conditions that the organism is screened under. And I can tell you, from about a deck of 35,000 compounds under 3 different conditions, so they're simple conditions. They're grown on glucose, grown on cholesterol, or grown on butyrate under slightly acidic conditions. If you compare those 3 sets of conditions, and you ask what's the top 100 compounds, there's 0 overlap, absolutely 0. So I mean, it is scary how much this bug can actually change its metabolism and adapt to different things, and how much that impacts the drug susceptibility. The lesion penetration, so we actually have a study I didn't talk about today in lung resection surgery patients, where patients get, at a defined time point, up to 24 hours, they get a bolus of 5 drugs, and they go in for their surgery. We take their tissue, section it, and do imaging mass spec in collaboration with the Novartis group in Basel, who've sort of pioneered this technique for looking at penetration of oncology drugs. And I can tell you the results are equally clear. I mean, some drugs get in incredibly well, and other drugs don't penetrate those lesions at all. There is some vascular defect in those. And so we've also been working with a guy at Harvard who studies angiogenesis, Rakesh Jain, who's done a lot in terms of understanding the vasculature and defects in vasculature in oncology. And a lot of the same story is true with TB. And we have lots of dysfunctional vessels. There's very poor perfusion of blood through those lesions. There's lots of anti-angiogenic consequences of being infected with TB. Whether that's a result of the bug or a result of the pathology, I'm not saying. That's the observation that you can see. It's quite clear. So I think the answer is both things. Probably to some extent, you've got to beat the ability of the drug to penetrate the lesion, but you also have to understand the physiology of the bug that you're targeting within that set of lesions well enough that you can pick targets that are ultimately active under that specific set of physiologic conditions. Tom?
That was amazing. I just think that nobody in the room could possibly look at their cultures or even their animal results the same way after that talk. I mean, it's really changing the whole field, and I really appreciate that you came today. I'm glad you didn't give the crystallography talk. So I do have a question, though. The premise was that the lesions resolve in response to chemotherapy at different rates. So in the sensitive patients, what are the lesions that resolve slowly? Are there increases in dose, for example, might be efficacious if it's penetration, drug penetration, something like that. Alternatively, if it's different physiology, as you sort of laid out, then we would need new targets for those lesions. That's the implication. Yeah. But what are the troublesome lesions in sensitive patients that would be the targets for a shorter regimen?
There's no surprises at all in this, right? And I mean, again, Denny Mitchison's a good friend and a big critic of mine. So he warmly points out that, what have you learned? You've learned that cavities are hard to sterilize. Well, I could have told you that 50 years ago, Clifton, saved you a lot of money and time. But I mean, there is no surprise. I mean, cavities are physiologically, pathologically, a really hard nut to unravel. I mean, lots of these lesions that look pretty bad on the CT, the lung can heal itself and rearrange pretty well. But cavities, you've actually lost that interstitial tissue. And I've never seen one go back to baseline, right? At best, they collapse, and they appear as a bronchiectasis, but they never go back to normal.
That sounds like it's a human physiology issue. And that maybe the bacteria aren't so different in cavities. It's just that's so severe a set of damages that it takes the body a while to ultimately respond. I mean, if it was an issue of penetration, for example, you'd expect drug resistance to be rife in areas where the drugs don't get in very well. And presumably we don't see that because you actually get cures.
That's in some extent, I think that to some extent that's true. To some extent, you do see drug resistance emerge in places where you wouldn't expect to see drug resistance emerge. And the bottom line is, if you sort of do the math and calculate, what the probability of drug resistance is by the individual mutation frequencies of each of the 4 drugs we use in frontline chemotherapy, you would need a biomass of bacteria larger than 10 human bodies to get a single resistant bug in one patient. So something more complicated is going on with that. And in individual environments, it may be that the pharmacodynamics are such that they're effectively only exposed to and selected for one drug at a time, and then that ultimately results in MDR or XDR disease. But it's not so simple. I'm not saying I have the answers, right? I just am pointing out what the holes are right now. And I'll also just say that every postdoc and fellow in my group who goes to South Korea and has this experience, and I try to get everybody through there, has the same, it is what Julie described in her talk. I mean, it's sort of the epiphany of this is why I do what I do, and this is why I have to do a better job in the lab.
So, very nice talk. I was wondering about your explanation. You showed the slide in which you didn't find a correlation between sputum culture, but yet, as you spun your stories from an anecdotal point of view, you were using time to culture positivity and finding sputum positive as relapse, and we've been using that, for 100 years. So I'm puzzled as to what's the explanation that you can, that you don't think that there's a correlation here. I mean, are we saying that there's colonization of Mycobacteria TB in sputum in high endemic areas, or are you getting reinfected with different strains and they're colonizing the sputum? So, or is all this still endogenous, from the person per se, and it's still the same strain? I mean, it seems like this is, so edify me, please. Yeah,
well, so I really can't give you a good answer to that, Larry. I mean, I'm quite cognizant of the fact that everything we have learned about sputum-borne bacilli in the last 5 years has been counter to what we thought about them in the previous 25. So there's a group in England, for instance, that's done transcriptional profiling of these. The traditional thought is they come from cavities that have eroded into the airways that provide an aerobic environment for the organism to multiply and go forth and prosper and infect somebody else. But in fact, every physiological characteristic we can see about the organism suggests that's not at all the case. They're not growing actively. They're not growing at all, in fact. And they look like they're dormant. They're dormant, what we think of in the laboratory as dormant organisms. And so I don't know what sputum's telling us. And there's no correlation at all between sputum positivity and the presence of cavities. There just isn't. I mean, it's an often quoted dogma with absolutely no evidence. And so they aren't coming from cavities. Where are they coming from?
You have a study that's so internally consistent, and then you do a meta-analysis when you have, different laboratory methods. Is the laboratory method itself the problem here, that it's sort of one of those true unrelated issues?
I think it's definitely part of the problem. Quantitative or even semi-quantitative bacteriology for tuberculosis has to be one of the most demanding microbiologic tests you can possibly do, and it's probably the highest standard of lab still has variable results. And so I think when you average across multiple labs, yes, you see that's probably contributing to that variability to a great extent.
Kim and Shalender, I have a question about the sputum analysis. I find it really interesting that you had this patient that was sputum negative. It's following up on this previous question, but yet the PET scans showed clear infection. So what I'm wondering is, there are new sequencing technologies that would enable people to take that sputum and actually look at not only what microbes might be present in different populations of microbes, like metagenomic studies, and that might be diagnostic, but also look at the genes that are expressed in the host, and that might be diagnostic.
Yeah, and we're not doing that, but I mean, we're giving samples to people for instance, David Sherman at the University of Washington has been doing some of that deep sequencing analysis from sputum samples with the GNF folks in San Diego, and we've been providing them samples, tissue, sputum, and other to do that kind of stuff. I think it's a very interesting approach, and definitely the idea that there are host biomarkers that might reflect when that relapse is going to happen is something that's very much in our minds, and I hope something like that exists, yeah. And the recent Ann O'Gara paper in Nature suggests that, right? Even in the peripheral circulation, you can see the difference between patients with active disease and latent disease, quote unquote, and then some of those latent patients who actually look like they're moving over towards active patients. So I mean, there's definitely a host transcriptional response that seems to be primarily this neutrophil-driven inflammatory response that accompanies active TB. So there probably is a good host signature there, but I think it needs some more work to work out.
All right, thanks, Cliff.
Sure.